that history. I love a good history lesson, and the timeline is really helpful and colorful. Um, can you tell us a little bit about what are the key assets within the Sentinel system? We know about the distributed network, which is really helpful in helping, protect patient privacy and to facilitate doing these queries quickly. But what kind of specific queries does Sentinel allow the FDA to do?
Sure. So, I mean, the data itself is one of the assets. And one of the reasons that it was needed to have 100 million people was really to look for rare events. I mean, if you're looking at something that's going to happen in 1 out of every million persons or 1 out of every 100,000 persons, you need a very large database to get to a statistically significant result associated with sort of any particular medical product. And so you really needed the data to actually capture the size, but then you needed tools or a way to ask those questions. And so we have been working for many years now to create a standardized set of tools. And I think there's a slide that Benzie has available to us to maybe take a look at to discuss what those tools might look like. But essentially, what we're talking about here is a set of computer programs. That's what we think of as tools. So they're SAS computer programs, and they're very sophisticated templates of how you might ask a research question. And they range from signal identification. So I have an exposure of interest, and I don't want to specify what outcomes I'm interested in. I just want to look at all outcomes. They also can be very focused between a particular medical product and one particular outcome. So I might want to study the risk, for example, in the Vioxx story, the risk of acute myocardial infarction following Vioxx exposure. And if I want to do that, I call that a level 1 query, and that's a descriptive query, and it's very similar to a standard sort of prep to research or prep to surveillance for a pharmacoepi study, where you're looking to understand what's the size of my study cohort, what are the most important characteristics of it, do I have enough sample size to do an actual study? And then those level 2 and level 3 that you see there, those are inferential analyses. So those are comparative analyses. They can be done with an active comparator design, or they can be done with a self-controlled design. And essentially, they're trying to answer the question, is my treatment group of interest, whoever that might be, more at risk for a particular outcome than my referent group? And those are really when you have a specific outcome that you're trying to target in mind, which is sometimes known as a signal evaluation study. It's a follow-up after you already have some suspicion that you want to sort of quantify what the risk looks like. It's designed to give a hazard ratio. And so it's a very traditional sort of observational study. I can do that one time after I've collected my entire sample size at the end of study, or I can do that in midway points throughout. And when I do that, that's known as a level 3. And that's when I'm looking at a first sort of bolus of new information. And really what I'm doing there is I'm checking to see, is there anything to be worried about here? And then I look again, and then I look again. And what that does is we do this while we control for overall type 1 error. So we make sure that we aren't giving ourselves more risk of having a false positive as a result of that. And this is a kind of, this was really what the concept of active was. I'm looking, I'm monitoring, I'm checking, and I'm watching essentially and seeing what happens as I go along. And so we've built out this sort of suite of tools that are available not only to the FDA, but also to anyone who basically has SAS and has a data set to run them on. So they're parameterized at the point of execution, they're all quality checked, and they produce a standard output. And you can see many examples of that on our website.
Yeah, that's terrific. And I'll just recall, that's an important point you just added there is the transparency aspect of Sentinel and the commitment to transparency and putting those tools up there on GitHub on the website and having them accessible to anyone to use. Now, the key is in implementing that, of course. But a nice description of the entire continuum of pharmacovigilance and pharmacosafety, all the way from signal detection and kind of those clusterings to really evaluating those signals against the comparator and over time. So tell us, like, we hear a lot about ARIA and how that is used, and the FDA takes on the action of safety surveillance for some products. Can you describe ARIA's efficiency and and what that is, and how does it get met, and if not, the determination that it is not met, and what is industry, pharmaceuticals, and biotech left to do?
Sure. So part of the legislation that created the Sentinel system in 2007 created this concept of sufficiency. And so what that means is that when the FDA observes information about a particular adverse event of interest that they think is experienced while on a medical product, they have to make a finding of whether or not the spontaneous reporting system and the Sentinel system, and particularly the ARIA system within it, are what's known as sufficient to study that problem. And if they find that the systems are sufficient, they actually execute their own study within the Sentinel system. If they find they are not sufficient, then that gives them the ability to require sponsors to do their own study, which is known as a post-market requirement. And so that's an observational epidemiological study that the FDA can require of a sponsor with certain conditions, usually involving chart validation, and then they're required to meet it. And so essentially then the sponsor is the one who is obliged to set up a study, create a protocol, et cetera. And so when the FDA does that, they also post their findings. So you can go on the website, there's a link here, and I think these slides will be available, and you can find out what did the FDA deem sufficient or insufficient to study a particular safety problem. And they've also started to include this information in the label. So there's a letter that sponsors receive when they have a product that's been approved. And in that letter, it will say, so in this case, there's on the screen an example of that. It will say that, the FDA plans to monitor this particular product in the Sentinel system as required by this is again referencing the 2007 Amendments Act. And it's saying that it found it sufficient, so you can see that wording in the space there, to basically study this risk of a particular outcome with respect to this spray. So if it finds this, and what the FDA is signaling then is that they intend to do this study. And they intend to actually look at this using the data, using the tools that they've developed. And so a manufacturer can look at this and they can say, oh, well, now I understand FDA intends to do this study. And they can also, likewise, in these letters, find it to be insufficient, where then they require the sponsor to do a study with some parameters, some specifications associated with it. And so that's what the sufficiency concept is around ARIA.
Thanks for that. And that's how I know that they come to IVED sometimes, because they are ARIA sufficient. So, but it's interesting to know, and I learned something new today, so is it that it is the spontaneous reports that trigger an assessment as to whether ARIA can be used to follow up a potential safety signal? Is that?